Alright, Bismillah. Let's start with doing a quick revision, I hope, for the upper GIT tract regarding the histology. Now, of course, if we're gonna start going with the starting with the histology for this video, it's gonna be starting with the oral cavity histology all the way to the esophagus. We're gonna take the stomach and duodenum in part 2, inshallah. And so we're gonna start with the oral cavity to the esophagus in that case. Tamam? Now, before we start, let's take a bit of an anatomy and physiology adding to our histology, so that it makes it easier for learning. Now, the number one is that when we go to the oral cavity, you gotta remember when food comes down to the oral cavity, it comes down in its most naked form. It's made of polymers and all that. And so your oral cavity with your teeth is to break down the food into its cellular level as much as it can, or closest to it, into basically carbohydrate, not carbohydrate, but basically tiny little bit of pieces of food with maybe some carbohydrate turned into glucose by salivary amylase enzyme secreted by your saliva. And the point is that they start to break down the food a little bit mechanically and maybe with some chemical secretion from saliva, and so that they can transport this food, let's say shawarma, into the pharynx and all the way to your esophagus till it reaches to your stomach. This is all part of the swallowing process, you could say. This is involving chewing and swallowing. So the point is how do we, knowing their anatomy and knowing their basic physiology, how does this apply to histology? Well, you got to remember, is it logic for your lips, for instance, and for your tongue, and basically, let's say the ducts of your salivary gland, your main duct, is it logic for them to be lined with something like simple columnar epithelium, those in the stomach and the duodenum? Of course not. They're not there to cause mainly digestion, or I guess you could say absorption. Okay, absorption. Their main goal is to break down some of the food and transport it down all the way to the beginning of your upper GIT tract. So it makes sense that the epithelium that they're lined by is going to be a durable epithelium known as stratified squamous epithelium or oral mucosa. Now, depending whether it's going to be a keratinized or non-keratinized doesn't really make much of a difference, but know it for the rest of this lecture as non-keratinized stratified squamous epithelium, which is a durable epithelium which protects against the abrasion of food. Abrasion is when you have like a chip and as it slides down, it starts to scratch off the mucosal surface. Yeah, that's what it's trying to protect against. Okay, so the abrasion of food with a bit of secretion of mucus, as we'll see later on. Okay, now, knowing with that idea in mind, let's continue with the oral cavity, starting with the lips. Now, your lips, Yazol, I like to use this picture. Now, your lips, they contain of 3 salivary structures. Just give me a minute. They consist of 3 not salivary structures, my bad. They consist of 3 covering surfaces, okay? 3 covering surfaces. You got the external surface, which is, yes, it is this part, just surrounding your lips. This part, the external surface, or your skin, which is lined by keratinized stratified squamous epithelium. And then you go deeper inside, you see the mucocutaneous junction. And the mucocutaneous junction, or in other words, if you read the books, vermilion zone, which is the reddish part, is known as the mucocutaneous junction. Muco for mucus, to relate to the oral mucosa, and cutaneous for skin. So we got the mucocutaneous region, which is the region in which there's a transition between the skin and the one that we're going to look into afterwards, اللي oral mucosa, your internal surface. We have to look at it from the inside. Like in this picture right here. So your skin overall has 3 surfaces, as we said. External surface, which is your skin, mucocutaneous junction, and the oral surface, or the junction between the surfaces, the mucocutaneous junction, okay? Now, if we're going to go internally, okay, and we're going to look at the lips, we're going to see that it has its internal surface, it's covered by an oral mucosa. And anytime you think of oral mucosa, you think of mucosa what? Epithelium and lamina propria underneath. Oral mucosa, that means the epithelium is non-keratinized stratified squamous epithelium and the lamina propria underneath. Okay, so epithelium here and a lamina propria underneath. And on the lamina propria of your lips contain a duct from a minor salivary gland. If you haven't noticed, or if you ever didn't know from before, we have major and minor. The major is what we took, the parotid and submandibular and all that. The minor are just little salivary glands that open into your oral cavity. One of them is called the labial salivary gland, okay? the labial salivary gland, in which it has its duct opening into the lamina propria of your oral mucosa. So another feature to keep in mind for the histology of the lips, as far as I'm concerned. And where is I guess you could say, where is the labial gland or the labial salivary gland? If this is their duct, they're found right underneath the lamina propria, right here, you could say, in the submucosa. And there's another picture that shows it, but basically they're found underneath the mucosa, on the submucosa, where they secrete saliva to ensure that your lips right here are kept wet. All right, so they're kept wet. And of course, you want to keep them where you want to drink water, right? Since the saliva is composed of 99% water, right? So prevent charred lips. So there's a couple of key features that we want to keep in mind. Now, this is, of course, this picture just shows us the oral mucosa. Let's look at it as a whole. Let's take a longitudinal section to further clarify what we're talking about. Like this portion right here, this beautiful picture. Now we got, like we said, the lips are containing of 3 covering surfaces. Internally, you've got the oral surface, which is lined by a very thick, keratinized, non-keratinized, stratified squamous epithelium. Thick, non-keratinized, stratified squamous epithelium. Now, as you go internally to externally, you'll see the vermilion zone or the mucocutaneous junction, which is the region in which there's a transition between the oral mucosa and the next one we're going to link it to the external surface, Elihian skin, which is lined by a thin, you can see the difference, thin, stratified squamous keratinized epithelium. So one is non-keratinized, one is keratinized. One is thick, one is thin. And like any other skin, it will contain hair follicles. It will contain hair follicles, sebaceous glands, salivary glands, the typical stuff that we would see under the skin, histology that is, right? And here is the one that we were talking about right here, this gland right here. So if we were to just remove these in a bit, we can see the presence of the labial gland found in the submucosa. And you can see the labial gland, they will have their ducts to secrete their saliva into the lamina propria of your lips, or of your oral mucosa, let's say, okay, to keep your lips wet. So that's the thing to keep in mind for the histology of the lips. Here is the same thing. Thick portion, transition to the mucocutaneous junction, and then from the mucocutaneous junction, it will transition to the external surface, that is your skin. All right, with the sebaceous and sweat gland hair follicles, and some mucus secreting labial glands underneath in the submucosa, okay? That's for the histology of the lips. Let's go to the next part. All right. So the next part is going to be talking about the histology of the tongue. We finished the lips, right? Knowing the 3 surfaces and which epithelium are they lined by. Now we want to look at the tongue. Now, of course, you know the tongue from the anatomy, consisting of 4 extrinsic and 4 intrinsic muscles, and they're covered by mucosa, right? Now, of course, it has 2 surfaces, right? The dorsum and the inferior surface. Let's look at the dorsum surface of the tongue only. Now, if you look at the dorsum surface of the tongue, now, as you know, before we start, the tongue is divided into 2 parts, right? The oral and the pharyngeal part, or anterior and posterior part, by that V-shaped sulcus terminalis. And you can see that the dorsum of the tongue is what? It's rough. It's not smooth. The surface is not smooth. It's rough, right? Now, part of that is because the posterior part of the tongue is lined by lingual tonsil. The other reason for the interior part of the tongue is because they're lined by 4 types of papillae. 4 types of papillae that contain, all of them contain your tasteless matter one. But before we go to that, what do we mean by papillae? Papillae, simply put, are basically mucosa, elevated mucosa. What do you mean by mucosa? Well, let's see. You see in the posterior part of your tongue, they're lined by mucosa. And we said what mucosa means, right? An epithelium, and underneath it, the lamina propria. Now, the epithelium that lines the posterior part of your tongue is the same as the anterior, which is what, like we said, in the oral cavity, non-keratinized, stratified squamous epithelium, right? With an underneath lamina propria. And the mucosa in the posterior part is normal. It's not elevated. However, when we look at the interior part of the tongue, we'll see that the oral mucosa that lines the muscles in the interior part of the tongue, the mucosa is elevated. And we gave it a name. And that name is papillae. And so we'll see that the rough surface of the interior part of the tongue, they're consisting of many different types of papillae, all right? Consisting of an elevated epithelium, non-keratinized stratified squamous epithelium, and a lamina propria underneath, Okay, 4 types اللي هم ايش خلينا نسميهم. One is anterior to your sulcus terminalis, your velate papillae. One is on the edges, you probably heard it for the first time, foliate papillae. They don't consider it in the anatomy because it's poorly developed in humans. They consider it as rudimentary, but still it's a part of the 4 papillae. So you got velate papillae, foliate papillae at the edges or on sides. On the edges, you've got fungiform or fungiform papillae. And the last one, the papillae that makes up, the papillae that makes up the majority of your papillae is your filiform papillae. Now, there's some things you had to keep in mind. Not all papillae, they contain taste buds. You see, the velate and the foliate and the fungiform, they all contain your taste buds, okay? They all contain your taste buds, except the filiform papillae. This is the only papillae that doesn't contain your taste buds for sensation. Instead, they just contribute to considering they make up the majority of your papillae, they make up the rough surface, they contribute to the rough surface of the tongue so that you're able to move food around as you're chewing. All right, so this is the only papillae that doesn't have a taste bud, but the remaining papillae, they do, even the rudimentary one, foliate. Now, if we want to go into, all right, so we know the taste buds are in these papillae, right? If we want to go a bit detail into, okay, what are the cells that make up the taste buds? Let's take a zoom in on the velate papillae and see what's going on. So you can see the velate papillae, it contains the taste buds within the epithelium, get a laterally-ish. And we can see the taste buds, they consist of 3 types of cells, all right? 2 cells are elongated, اللي هم supporting cell, which we don't know its function. And the other one is your gustatory cell, which is the same as saying sensory cell. Don't worry, it's the same thing. And this is the cell that's mainly responsible for picking up chemical signals from food and basically tell our sensory nerve, which would be the chordae tympani nerve, to send a signal to the brain to get the sensation of taste. Okay, so that's your sensory cell, your supporting cell, and the third cell that make up your taste bud. And like I say, bud معناها empty sac. The last cell that makes it up is your basal cell. Okay, those are the cells that make up your taste buds, which again are located on all papillae except filiform papillae. All right, so that's something to keep in mind as well. Now, of course, another thing to keep in mind, are the taste buds only located in these papillae? No, there's other structures as well, like the pharynx, your epiglottis, and your soft palate as well. We're going to look into them, but if you want to get an SCQ says enumerate 2 locations of the taste buds, maybe you could choose these answers instead if you're paranoid about the spelling, all right? So that's for the papillae and the dorsum surface of the tongue. Obviously, before we move on with the tongue, another thing to keep in mind, and of course, here's the histology of the taste buds, but before we move on, I want to look into basically something very important in one of the papillae. And that something very important for one of the papillae is circumvallate papillae. You see, in the circumvallate papillae, because we're going to learn all about this in the OSPE, but I just want you to know that in one of the papillae, alias circumvallate papillae, they contain a very special minor salivary gland, all right, that secretes serous saliva called von Ebner glands or von Ednar glands. Von Ednar glands in their lamina propria, okay, or in their submucosa, either way, in the lamina propria, let's say. Now, these von Ednar glands are minor salivary glands that are found in only the circumvallate papillae, maybe foliate, but mainly in the circumvallate papillae, okay? And the main function of these von Ednar glands is that being saliva, they secrete saliva through their ducts, and their ducts, they terminate into these clefts, these empty spaces between the large circumvallate papillae, to ensure, and of course the reason why is to ensure is that if you have any food that come down to the clefts, they could overstimulate your taste bud over and over again. So you keep tasting the same apple you ate a minute ago over and over again. What they do is they secrete saliva into these clefts to wash them away, to wash all these food particles away, so that instead of tasting the same apple over and over again, you could taste, for example, a banana next. Okay, so it's like a washing cleaner of sorts. So that's just something to keep in mind as well. Very important. So there could be an MCQ. They could be asking you which papillae of the tongue contain stratified squamous epithelium and von Ednar glands in its lamina propria. You will choose circumvallate papillae, which are these 2 darker structures as well. Okay? So that's something to keep in mind. They are minor salivary glands, mind you. Okay? And that's basically what we talked about. The 4 types With the exception of filiform, they all contain taste buds. And we said the cells that make up the taste buds. Of course, here's the circumvallate papillae. Again, the von Ebner glands. Again, they're only found in circumvallate papillae. And we said their purpose through excretory ducts. Secrete saliva, washing away food particles. Here's another structure showing us the ovoid structure known as the taste buds within the non-keratinized stratified squamous epithelium. Remember, the oral tongue is lined by oral mucosa, so it's immediately lined by that type of epithelium. Again, to protect against the abrasion of food. And of course, the last one is the location of your taste buds. Now, I don't want to go into more detail about the histological feature of each papillae ma'ada circumvallate because we're going to know more in the others in the OSPE. But for now, we should know one absolutely for the circumvallate, for the finals. I mean, for the mid-exams and the finals too. Now, like we said before, are the taste buds just located on the papillae? No. There's other structures that I mentioned before where they also have taste buds. We have the soft palate, the pharynx, and the epiglottis. Okay, so that's something to memorize as well. Enumerate 2 locations of the taste buds. You can answer any of the 2. All right. So that's the microscopic feature of the tongue, and of course the posterior one-third of the tongue lined by the same epithelium. It's just it's not like elevated. It doesn't have papillae. Just lingual tonsils like in the interior. The posterior one-third of the tongue contain just oral mucosa, stratified squamous epithelium, and basically it. We're not going to focus into it too much. Just know the entire tongue is lined by non-keratinized stratified squamous epithelium. And that's it for the tongue. Let's go to the salivary gland next. All right. Now, when it comes to the salivary glands, now up to this point in anatomy, you've only known the major salivary glands, right? The parotid, the submandibular, and the sublingual, which secrete not all saliva, 90% of saliva. The ones that we haven't taken much notice, they're not that important, is the minor salivary glands. I gave some examples like buccal, labial salivary glands, the one that we've seen before in the lamina propria of the lips, right? And the palatine, for example, they secrete the remaining 10% of saliva. So when we look at the histology, we're not going to focus on the minor. We're going to focus on the major parts, okay? But this is something to keep in mind that we don't just have major, all right? So if we want to look at the histology of the parotid and submandibular and sublingual gland, these exocrine glands, we have to first summarize it in a beautiful picture, all right? So something to keep in mind as a principle is that any exocrine gland, by principle, has to be covered by a capsule, all right? It has to be covered by a capsule. Let's imagine this is an exocrine gland, like a salivary gland. They have to be covered by this blackish color, a capsule, a dense, irregular connective tissue capsule, all right? So, and arising from this capsule is a part of its stroma. Any gland has a stroma and parenchyma. So, arising from the capsule are going to be these septa, which are walls, which are like this. They're extending from the capsule, all right? Extending from the capsule. So, again, the exocrine salivary glands, they're surrounded by a capsule, okay, made of dense, irregular connective tissue. And extending from the capsule are these walls. We call these septa, or in plural, septum. And basically, you see this empty space right here? This was the parenchyma. Parenchyma meaning the functioning part of the organ, while the capsule and the stroma, they were the supporting part of the organ, making up the stroma. So this empty space right here was basically your parenchyma. And the septa that was arising from the capsule, they were dividing the parenchyma into lobules, all right? Into lobules. So they were dividing them into lobules, like here and here and here and here and here. And within these lobules contain 2 important things that make up your salivary glands, or your not your kidney parenchyma, your salivary gland parenchyma, okay? Number one, and hold on, let me just, now let's draw it, let's put it right here on top of it so you can understand it, because we're going to need it later on. So the lobules that make up, or your parenchyma consisting of these lobules, they make up of 2 things, your secretory acini and your ducts. So let's divide them. So your lobules, right here, your lobules are going to be composed of, the one side is secretory part of the gland, and the other one is going to contain glandular ducts, okay? Glandular ducts. Now, one thing to keep in mind, we're talking about the main structure of all 3 main parts of the salivary gland, the parotid, then the submandibular, and all that. Now, I just wanted to keep you in mind that the The difference in all of them is going to be in the secretory cells, the cells that make up them. But all the glands are going to have the same ducts, as we are going to see shortly, okay? So again, back in topic. The lobules are consisting of a secretory part and a glandular duct system, okay? Now the secretory part are divided into 4 types of cells, basically. What are these 4 types of cells? You got the first one, serous cells. All right, serous cells. From their name, they secrete saliva, watery saliva rich in proteins and enzymes, which is what the parotid gland is made of exclusively. The second one from the secretory part is your mucus cell. From their name, they secrete sticky, a very thick saliva, right? Rich in mucin, a glycoprotein that is found not in parotid but in the other sub, okay? The third type, and we took it in repro for the breast, is called myoepithelial glands. Myoepithelial cells, my bad. Myoepithelial cells. Myo معناها muscle. Epithelial معناها epithelial. So these are specialized epithelial cells that surround your mucus cell and your serous cell, and from their function is that they contract. When they contract, their function is to push the contents of the saliva, the serous and the mucus saliva, into the duct. All right? That's their basic function, essentially. We're looking into it in another drawing. But we got myoepithelial cells. And the last one is a mixture, it's a combination of the mucus and the serous cell called the serous dimulans. Dimulans معناها concentric in shape. يعني إيش؟ يعني معك كده mucus cell, معك كده mucus cell, and you got these mucus cell joined together to form tubules, tubules. And then you've got the serous cell. You've got the serous cell right here, just attaching to it or covering it, capping it. And you form the serous dimulans, which is like a concentric shape, a half moon. All right? And these are only found in the submandibular and sublingual, like we'll see later on. But the main point is the secretory parts of your glands are going to consist of serous cell, mucus cell, myoepithelial cell, or a combination of mucus and serous called serous dimulans. All right? That's going to be the difference in all 3 glands. Now let's look at the glandular duct system, shall we? Now the glands, the ducts that make up your salivary gland are going to be the same. So you've got, you've got number one, your intercalated duct. Your intercalated duct, that's the first duct that all the serous and the mucus cell, they're going to secrete their contents into, these intercalated ducts, all right? Which are lined by simple cuboidal epithelium. Then after the intercalated ducts, these intercalated ducts are going to go to another duct. They're going to drain saliva into another duct. Basically, intercalated ducts are going to join with other intercalated ducts into another duct that's striated. It is called striated duct. Okay? Striated duct. From their names, they're lined by cells that are striated. What is the meaning of striated? They have streaks. They have streaks like these. And that's as a result of their mitochondria, parallel to each other, vertically oriented. And these striated ducts are lined by basically striated columnar epithelium, okay? So your intercalated ducts, they're lined by which type of epithelial cells? Simple cuboidal epithelium. Then the striated duct, we come to the striated duct, they're lined by what? Striated, and was that striated even? Simple columnar epithelium that are striated in appearance, okay? Now these two ducts together, if we were to draw it for you, all right, if we were to draw it, these 2 ducts, like let's say we have the intercalated ducts right here, and they drain into the striated duct, okay, and this striated duct will then move on, all right, in the black color. These 2 ducts, the striated duct and the intercalated duct, they make up the intralobular duct. What is it called? What is it called again? Your intralobular duct. What is the meaning of intralobular duct? Meaning these ducts, like imagine these are your lobules, all right? These are lobules, meaning these ducts are present within the lobule. They come directly from the serous and mucus cells, but they're present within the lobules. Now these intralobular ducts, these intralobular ducts are going to be draining into the next duct, okay? Which is called interlobular duct. Not intra, interlobular ducts. And from their names, these interlobular ducts, they're the ones that are going to be found in between the lobules. So we're going to draw it in a blue color. It means the striated duct is going to join with the other striated duct to drain into the main, not the main duct, but the duct that is found in between each lobule, اللي هم interlobular duct. Okay, found in between each lobules. All right, that's the meaning of interlobular, found in between the duct found in between each lobules. Now the interlobular duct is quite complicated, the epithelium. I'm not gonna write it down because it's long, but basically know it that the interlobular ducts, they consist of many types of cells. As a result, their epithelium is going to be different, and their epithelium that lines them is like this: pseudostratified or stratified, columnar or cuboidal epithelium. You get that point? I hope so. They're lined by pseudostratified or stratified, columnar or cuboidal epithelium. Many of these different types of epithelial cells lining your interlobular duct, okay? Now your interlobular duct is not your main duct. It's not the final duct. If you notice, like let's say imagine right here, let's put it in the blue color again. Your interlobular ducts, yeah, there are the ducts found between each lobules, but they're not going to be the main duct that's going to be coming out of the salivary gland. These interlobular ducts are going to drain into the final duct, the main duct, and it is called your main duct, coming out of the salivary gland, all right? Salivary gland. In other words, the main duct could also be called for the parotid gland, parotid duct. Your main duct in submandibular duct. So submandibular duct, sublingual duct, parotid ducts, the one that come out of their glands respectively, those are your main ducts that they're talking about, okay? So this is your main duct, the final duct that consists of the glandular duct system. Which epithelium is your main duct lined by? It's very easy. It's lined by non-keratinized, stratified squamous epithelium. Okay, non-keratinized, stratified squamous epithelium. That's the final duct coming out of the salivary gland. Okay, your main duct to terminate whatever, right? The oral vestibule for the parotid gland and so on. So this is your glandular duct system. And the glandular duct system, again, is the same for all three glands, major glands, parotid, submandibular, sublingual. The difference is going to be in the secretory part. Your parotid gland is going to be consisting exclusively of serous cells, okay, and myoepithelial cells. Your submandibular and your sublingual, they're going to contain the serous dimulans, which is the mixture of both mucus and serous, okay? So they're a mixed gland, while the other is exclusively serous. So we're going to look into them later on. But this is, in summary, the histological arrangement of your salivary glands, okay? And that's the entire point of this entire slide, okay? And the slide over here as well. Of course, we didn't go much into detail. Of course, like the mucus cell, they all join together to form tubules. All right, and the serous cell, they join together by tight junctions to form acinus. Acinus means spherical structures, okay? So you got mucus tubules resembling tubules, serous resembling acinar structures. They're pyramidal in shape, the typical stuff. And we can see, like we said before, the serous acinar structures and the mucus tubules. They all drain to the intercalated ducts, and then the intercalated ducts to the striated duct, then the striated duct to the interlobular ducts, and the interlobular ducts to the main duct. And you know the epithelium that they're lined by. Tamam? And we can see also, let's not forget, the serous dimulans, other structures as well, mixed acinas, and the myoepithelial cells, which are lined, like we said, they're surrounding the serous and the mucus cells, specifically in their basement membrane, as you can see. They're basically flat, all right? They're basically flattened cells, all right? Or squamous-like cells, all right? So that's basically it for the epithelial components of the salivary glands, like in general, taban, okay? In general, we've seen these as we said before. And of course, here's the glandular duct system. I want you just to look with that we've said before what they are. And of course, here's the epithelium of your interlobular duct. Again, just memorize it like it is. Pseudostratified or stratified, cuboidal or columnar epithelium. Tamam? Epithelium. So that's basically it for the main parts of the salivary glands. Now we want to go into specifics for each gland now. All right? For each gland. The parotid, the submandibular, with sublingual gland. Now, let's start with the parotid gland. Now, your parotid gland, they secrete saliva, right? But is it of serous or the mucus only serous acini. So if I were to describe you the parotid gland, it's an exocrine gland. It's a branched acinar gland that is made of exclusively serous cells. Thus, they secrete only what? Serous saliva. So if you look at this histological picture, what do you see? You see these darkened cells right here? These are your serous cells, which appear darker in stain. Why do they appear darker in stain? Because they contain mini granules, rich in enzymes and proteins. That's why. So your darker stained cells right here, just darker stained cells in your gland, this is parotid gland. And now before you ask, this is not mucus cell, okay? This is most likely striated duct, okay? So we can see the presence of serous cell right here and some striated duct as well. And of course, before we go, if you don't remember the foundation histology regarding the structures of exocrine glands, yani simple, branched, tubuloacinar, and acinar, this is a picture that illustrates. Branched acinar, branched manaha, the duct is going to be branching out, the branching of duct. So when you say branched, that means the duct are branching out. Branching out into what? Acinar structures. Hence the name as branched acinar or alveolar, the same thing, okay? And of course, when we say acinar, meaning these are containing serous cells, serous acini, okay? That's the meaning of it. So they're only branched acinar only, not tubuloacinar, okay? And they contain the same ducts as we said, okay? Easy. So that's the parotid salivary glands. Go to the submandibular gland. Now the submandibular gland, it is not made of exclusively serous cell. Okay, just like the sublingual. So they're not made of serous cell. They're made of both serous and mucus cell. And if we look at this picture, we can see the presence of the darkest stained cells that we mentioned before for serous cell, right? Mini granules. But we can also see the presence of mucus cell, which appear pale because mucin stains pale in their H&E stain, okay? So you see the presence of these mucus cells and the presence of these serous cells. So in that case, this is either submandibular or sublingual. Why do I say either? That's because the only difference between submandibular and sublingual histologically is the population between serous and mucus. What do I mean? I mean, for example, your submandibular gland, let's say they contain 60%, let's give an imaginary number, 60% serous and 40% mucus, okay? They're a mixture of both. They're a mixed gland. They contain both serous and mucus. However, the majority of cells that make them up is serous, since they predominate. That's why in this picture, if you see both serous and mucus, but the majority of these cells predominate are serous acini, this is submandibular and not sublingual, because sublingual is the opposite. Okay, it's the opposite. That's why in some books they say sublingual secrete only mucus. It's not true, but they mean the majority of them secrete mucus. Okay, that's the meaning of serous acini predominate. And again, if you don't remember foundation histology, according to exocrine gland structures, we said they were what? Branched, They were branched tubulo-alveolar for the submandibular and the sublingual. Branched, we meant the ducts. They branch into what? They branch into tubules, and acinar. So you got a acinar right here and you got tubules. So branched tubulo-acinar. And we said tubules, they represent your mucus tubules, and your acinar represents serous acini. So it's a mixture of both, like we said. So it's a branched tubulo-alveolar or tubulo-acinar glands that consist of both serous and mucus. But which one is more serous acini? They predominate. And we could say, instead of saying serous and mucus, I'm going to say serous dimulans. Okay? With the same ducts as we mentioned. Here, the serous dimulans, like we said. Here's the serous acini covering the mucus. Here's another picture. Here's your serous acini, and this is your mucus, pale stained under electron microscope. Okay? So that's pretty much it for the submandibular, and that's pretty much it for the sublingual as well. Because the sublingual is the same thing. The only difference is that let's give it an example. Let's say right here, for example, 90% of it contain mucus, acini or tubules, and 10% serous. Okay? So as a result, when you look at histologically, you'll see many serous dimulans, but the majority of them are what? Mucus tubules in the sublingual gland. Mucus tubules, with some serous acini capping them, forming the serous dimulans. All right? That's the only difference. Mucus acini predominate in the sublingual gland. And again, it's a branched, meaning ducts branching into tubules and acinar. Hence the name branched tubulo for mucus, alveolar for serous. Branched tubulo-alveolar glands. Okay? And they consist both sublingual, submandibular, serous dimulans. تمام. That's it for the salivary gland. Just as easy as I've predicted. All right. Now we finished the sublingual and the submandibular and the parotid gland. Now let's look at the GIT tract, the esophagus. Now, when it comes to the histology of the esophagus, it is the easiest. I mean, it's easiest because it's a revision of the physiology lesson number 1. If you go to physiology, the overview is literally the same. Mucosa, submucosa, musculus externa, in which it mentioned that the layers of the GIT tract, they were made of 4, right? Mucosa, submucosa, muscularis, and adventitia or serosa, depending. And it said it's starting from the esophagus to the anus. They all contain the same 4 sublayers, but with some different characteristic features. Like we're starting with the esophagus, all right? So when we go down, you'll see the 4 layers, very easy. There's going to be a difference, for example, in their epithelium. Starting with the esophagus, all right? So if we're going to start, let's just revise basically the physiology. Now remember, you remember the esophagus, Yasor? You remember the esophagus, which is this long muscular tube? Tell me, do you think this makes sense for esophagus to be lined by simple columnar epithelium? No, the esophagus is not meant for digestion or absorption. It's exactly like you see it. It's a muscular tube whose purpose is literally to transport a bolus from the pharynx into the stomach. Thus, the epithelium that it should be lined by should be an epithelium that is durable, resist the damage from the abrasion of food. However, it's not as protective against acid, unfortunately, like you see in GERD. But the epithelium that lines the esophagus for protection and for secreting mucus too, not for stomach acid, mind you, for the abrasion against food, is stratified squamous. The epithelium that lines it is stratified squamous epithelium. Keratinized or non-keratinized? Non-keratinized stratified squamous epithelium. That is the epithelium that lines your esophagus. Logic, all right? So if we go to the esophagus, that's the difference immediately. That's the difference immediately from your esophagus to your, let's say, your stomach and intestine, is that they're lined by an epithelium that is not found in the stomach, okay? Because in the stomach and intestine, we're going to see they're lined by simple columnar epithelium, for instance, okay? So that's immediately for the esophagus, which is divided into 4 layers. We said the mucosa is lined by what epithelium? Non-keratinized, stratified squamous epithelium. And mucosa, meaning epithelium, was a lamina propria underneath. Revising for physiology, it also contains galt. Okay, the lamina propria is a thin, loose connective tissue containing galt, which stands for gut-associated lymphoid tissue. If you remember from physiology lesson 1. And underneath it is muscularis mucosa. This muscularis mucosa is a thin layer of smooth muscle responsible for movement of the mucosa, basically for helping in movement of the mucosa that it secretes, the mucus that it secretes, that is. Okay, so you got the muscularis mucosa. That is the mucosa of your esophagus, just like that. Now, what is underneath the mucosa? Your submucosa, taban. Because we just finished here. This is, of course, the small intestine, but you can imagine the esophagus without being folded, all right? Now, let's go to the submucosa. Now, here's the cool thing about the submucosa. It is a thick, dense, irregular connective tissue, okay? A thick, dense, irregular connective tissue. This is basically the tissue. This layer right here is the one that's responsible for giving any digestive tube this tensibility and elasticity. Meaning what? It means basically when food comes here, it basically makes sure that your intestine or your esophagus doesn't explode, all right? It's able to be distended, stretch it. But also make sure that's not permanently distended. It can be elastic, return back to its form once it pushes the food forward, propel it forward via peristalsis. So that's what the submucosa does. It contains this dense irregular connective tissue responsible for that. So the submucosa. And there's another exception you need to know for the esophagus. The esophagus, it is the only upper GIT along with the duodenum that contains glands in their submucosa. They contain submucosal glands. The duodenum and the esophagus, they're the only organs or the only digestive tubes in your upper GIT that contain glands in their submucosa. And those 2 glands that they have are basically mucus-secreting glands. So in the case of the esophagus, the submucosal gland in the esophagus, I'm drawing on the small intestine, but you can imagine the esophagus, right? But in the submucosa, the gland that they have is called esophageal gland. They also have another gland, but we're not going to look into it. They're called cardiac glands, but we're going to focus on the esophageal gland. The esophageal glands, from their name, they're mucus secreting gland. They secrete mucus so that when there's food coming down like a chip that you haven't eaten properly, it makes sure that the sharp edges of the chip doesn't damage doesn't cause erosions of the esophagus by secreting mucus, making it, lubricating it. Okay, they come from the esophageal glands located in the submucosa. And like any other digestive tube, they contain a nerve plexus as well called the submucosal plexus. All right, so in the esophagus, they contain, along with the duodenum, they're the only ones that contain a gland in their submucosa. For the esophagus, it's called the esophageal glands, secreting mucus for protective and lubricating purposes. The other thing they contain is like any other digestive tube, اللي هي submucosal plexus. All right, that's the most important parts in the dense irregular connective tissue. The next one underneath it is the muscularis layer. Now, the muscularis externa layer, this is the layer responsible for contraction. Therefore, peristalsis and mixing movement. They contain of two layers. One layer is basically circling the lumen, known as the inner circular layer. And the other one is a layer that is lining the whole length of the digestive tube, calling the outer longitudinal layer. And in between these sublayers in the muscularis layer is the myenteric nerve plexus. My for muscle, enteric for intestine. So a nerve plexus found from their name within the muscularis layer of a intestine, all right, or within a digestive tube, okay? Now, I want you to keep in mind, when it comes to the esophagus, they actually contain both skeletal muscle and smooth muscle. So we cannot just say inner and outer longitudinal smooth muscle layer. No. Because your esophagus, they contain both depending on the region. So if I were to draw for you the esophagus, simply like here, the upper part of your esophagus, they're going to be lined by exclusively skeletal muscle. If you remember from the lesson 1 on physiology, they mentioned that motility is achieved by smooth muscle contraction except 3 parts. One of them was the early part of the esophagus, which was lined by skeletal muscle. So the upper 1/3 of your esophagus lined by skeletal muscle. The middle is lined by a mixture of both skeletal muscle and smooth muscle. And the lower end of your esophagus is gonna be lined exclusively of inner and outer longitudinal smooth muscle layer. Again. So again, the esophagus is lined by skeletal muscle and smooth muscle layer, depending on the location. The upper end is exclusively skeletal muscle, okay? Exclusively. Lower end, exclusively smooth muscle layer. In the middle, a mixture of both smooth muscle and skeletal muscle layer, okay? So that's something to keep in mind. So that's for the muscularis layer. And now the final part, the most outermost layer is the serosa. Now, not just the serosa for the esophagus, but the adventitia. Let's look at it from a slide real quick. But for now, we're going to look at it in a slide of its own. But for now, you get the points, right? The mucosa, the submucosa, the muscularis, the features of not just our digestive tract, but the esophagus as well. All right? So let's revise it, what the esophagus contain. Of course, these are identification points for the OSPE to keep in mind, according to what Dr. Nimmer has said, for the OSPE to write in. So we get, again, the mucosa of the esophagus, they're lined by what? Non-keratinized stratified squamous epithelium, with a lamina propria and a muscularis mucosa, sublayers. All right, like we can see right here, very thick stratified squamous epithelium. And here is the submucosa. If this is the mucosa, right, with the epithelium, the lamina propria and the muscularis mucosa, here is the submucosa, and you can see the presence of esophageal glands with excretory ducts. They're the ones that secrete mucus, to protect against the chip, for example, here, because the abrasion of food. So you can see these mucus acini of esophageal glands. They're the only ones besides the duodenum that have these in their submucosa. Okay? And of course, the muscularis layer, depending on which region, skeletal or less smooth muscle. Here's the outermost layer we're going to look into, the adipose tissue and serosa. They're lined by both, mind you, but depending on the location. Here's another picture as well. Easy, certified squamous epithelium, non-keratinized. And here's the submucosa. The most important thing you need to know, they contain the esophageal gland with their excretory ducts and the submucosal plexus. And by the way, the submucosal plexus and the myenteric plexus that we're going to look into here in the muscularis externa, these together form your gut brain. They form your enteric nervous system, okay? As you know from lesson 1 of physiology, the overview that is, all right? So dense irregular connective tissue, and here's again, remember esophageal glands, تمام. Was the muscularis layer. Here's the muscularis external layer, inner and outer longitudinal muscular layer, depending on the region. In between them is the connective tissue containing the myenteric plexus, as we can see right here, connective tissue containing them. Okay. And finally, we'll go to the outermost layer, serosa with adventitia. Now, your esophagus, they contain both adventitia and serosa, but depending on the location. So here's how you're going to draw it. You see here, we're going to draw here the esophagus, okay? Your esophagus is going to be lined by adventitia. The majority of it, they're all going to be lined by adventitia, which is a loose connective tissue, except the distal, the lower third of your esophagus. They're going to be lined by serosa. So, hold on. So, basically, your esophagus is lined by adventitia and serosa. All the parts of the esophagus, all of them are lined by the adventitia, the loose connective tissue, معدا the distal كده 1 to 2 centimeter, يعني the lower third of your esophagus, which is lined by serosa. Why? Because the lower third of your esophagus, they're present within the abdominal cavity. You see, anything that's present within the abdominal cavity, when they're squished around by digestive organs, they need to be lined by serosa. And you know the function of serosa, right, from physiology. Secretes serous fluid, so they can provide lubrication to prevent friction. All right, so that's why they're lined by serosa. But with the adventitia, on the other hand, lining your esophagus the majority of the time, their purpose is to basically connect your esophagus here into the stomach. All right, so that's the function of your adventitia, to literally attach your esophagus to the stomach. But the serosa, its purpose is to prevent friction between the esophagus and the stomach. All right, found only in the lower third of your esophagus, there's serosa. The majority or the remaining amount in your adventitia, or lined by adventitia. Okay. And the last thing just to keep in mind is, of course, the serosa. They are the connective tissue that is. It is lined by an epithelium, a simple squamous epithelium. We gave it a special name called the meso or the mesothelium. All right. Another feature to keep in mind for the serosa. Okay. They're both two loose connective tissue, but with two different functions. One is basically attach the esophagus to the stomach. The other is for lubricating purposes. Okay. One has an epithelium covering it. The other doesn't. اللي هي الادفنتيشن. Okay, الميزوثيليوم الكلام ده. And that's basically it. Like here, like we said, the upper esophagus, only adventitia. All right. Here in the lower esophagus, what are you going to find? Serosa. You see the difference now? You see the difference? Here's another part. Here's the thing I was talking about the muscular layer. The upper esophagus, the skeletal muscles. But the lower esophagus, smooth muscle. Okay? Depending on the region you're talking about. And here's the gastroesophageal junction we're talking about. And here's the esophageal glands. All right, they're the only ones that have glands in their submucosa, which has a gland called Burner glands. Memorize that name for when you look at part 2. Okay? And that's the end of histology. Knowing the 4 layers of the esophagus, you see some key differences, right, between the esophagus and the other remaining digestive tubes. All right? Good luck in your revision, and thank you.